Welcome, everyone. My name is Cora Vacher. I'm the Associate Director for Human Genetics in Strategic Marketing at Oxford Nanopore Technologies. Thank you very much for joining this knowledge exchange today. We will be looking at how Oxford Nanopore software solution, together with our partner, help overcome the challenges of data analysis for rare disease. After the talk, we will have a live Q&A session, so please add your question to the Q&A box, which you'll find at the bottom of your screen. You can also upvote the question you would most like answered, and we will try to get through as many as we can. This knowledge exchange is being recorded and will be made available on demand after the broadcast. During this short introduction, I'm going to cover how discovery and prioritization of variants in rare disease can empower clinical research. So we all know rare diseases are not rare. They affect more than 300 to 400 million people worldwide. At the level of the individual, a rare disease affects fewer than 1 in 200 individuals, but there are over 8,000 rare diseases identified so far. Some can lead to very severe multisystem disorder, but many of them still display a very wide range of phenotype, making the characterization of the disease based on phenotype only quite challenging. 80% of rare disease have a genetic origin, and therefore, genetic testing in clinical genetic lab has been implemented as part of the toolbox for routine standard of care. There's still a lot of hidden genetic variants, non-contributor to the disease, that are missed by the routine genetic screening method. If you look at coding region, almost 3,000 non-coding regions are hidden in plain sight. This can be either because the chemistry from the short read method is not suitable to generate the sequence, or if the sequence is generated, it can be because due to the short length, it's very difficult to analyze it and assemble it. We know that 34% of all disease-causing variation is made up of variants that are larger than a single base pair substitution. And those structural variants have a much likelier impact, 30-fold in fact, to have an effect on gene expression compared to SNPs. If we look at even larger region, complex chromosomal rearrangement involve very large genomic segment, sometimes several megabases in length. Last but not least, methylation plays an important part in rare disease, in particular in imprinting disorder. And standard methylation arrays only cover less than 3% of all the CpG sites in the genome. So when we look at the most routinely used methods in clinical genetics, we can see the leaf gap, whether it's Sanger sequencing, PCR, microarray, or next generation sequencing with panel, short-read exome, or short-read genome. If you look at the list of variant type on the left, you can see that some methods do not cover those at all. I'm thinking in particular gene and pseudogenes, mosaicism, and of course, again, methylation. With Oxford Nanopore sequencing, you have one assay that enables you to capture the information on all of those variants. There is a mounting amount of evidence that long read really enable resolving challenging cases. I'm thinking in particular compound heterozygous, where phasing is really important, but also regions that are rich in tandem repeats or repeat expansion, and copy number variant, where the sheer length of the reads enables the physical coverage of the region and its resolution. At Oxford Nanopore, we do have a solution from sample to answer. This covers preparation of your DNA or RNA in a manual or automated way. Of course, we do have devices that are low to high throughput, and Jennifer will cover in depth the analysis. But I want to point out that it's very accessible, scalable, and we have a very versatile portfolio of analysis. So we can take your data analysis and interpretation much further. Through EPI2ME, we do have human variation workflow that enables secondary analysis even when the lab doesn't have a lot of bioinformatic capabilities. It's very user-friendly and doesn't require coding. EPI2ME is now integrated with third-party analysis that enables tertiary analysis and even in the future, full interpretation of the variant. So in short, with Oxford Nanopore sequencing, you will have the same information that you would have with short read. Single nucleotide variant, and of course, insertion and deletion. But you will also have a lot more information, including structural variation, copy number variation, and complex structural rearrangement, and of course, not to forget methylation. Now, I'd like to welcome Dr. Jennifer Griebel, Field Application Scientist in Bioinformatics at Oxford Nanopore Technologies. She will present an overview of human variation workflow, and then we'll focus on data analysis, on the data analysis part through EPI2ME. Over to you, Jennifer.
Thank you so much for that introduction, Cora. As mentioned, my name is Jennifer, and I am a bioinformatics field application scientist with Oxford Nanopore Technologies covering the central United States. I'm going to share with you an overview of our premier end-to-end bioinformatics solution for the detection and quantification of variation in the human genome. Oxford Nanopore provides an ecosystem of software tools that create data analysis solutions for all levels of expertise. When you perform sequencing, you interact directly with the Minnow application, which controls the sequencing device, generating raw sequencing data, and performs some initial analyses. Once you have generated your sequencing data, you will need to analyze it. EPI2ME is our bioinformatic analysis platform that is accessible to scientists with all levels of expertise. EPI2ME's pre-configured workflows can be utilized as a desktop application on any computer, with options for both local and cloud-based analyses. EPI2ME's bioinformatic workflows can also be implemented via the command line to deploy on a high-performance compute environment. Bioinformaticians can find Oxford Nanopore's latest tools and algorithms on GitHub to implement in your own custom pipelines. The Oxford Nanopore sequencing platform has been consistently elevated by the engagement from our community members, and so if you are interested in a particularly niche application, we recommend that you search through community-developed tools or even write your own. We offer support for Minnow, all EPI2ME workflows, and Oxford Nanopore developed GitHub tools. Today, I'm going to focus on the EPI2ME application and the workflows it houses. EPI2ME is Oxford Nanopore's fully supported desktop application that provides a range of pre-configured workflows, including a comprehensive human genome variation workflow. The benefit of utilizing EPI2ME is that it provides intuitive and interactive reports, as well as standard bioinformatic output files. EPI2ME's workflows can be run locally or in the cloud. These workflows are also available to run directly on the command line for flexible deployment of your analysis pipeline. EPI2ME is able to integrate with your own custom pipelines as well as tertiary analysis software. Nextflow pipelines that follow the NF-Core format can be imported into the EPI2ME application for deployment across your organization. Moreover, you can connect EPI2ME directly with tertiary partner software. We currently support several software focusing on prioritizing variants within the space of rare and undiagnosed disease. EPI2ME offers a pre-configured end-to-end workflow for the comprehensive analysis of variation in the human genome. These variants include single nucleotide substitutions, indels, large structural variants, copy number variants, and short tandem repeat expansions. The EPI2ME human variation workflow also quantifies modifications. Data will be input into the workflow following base calling. Once complete, the EPI2ME analysis provides annotation files and reports for all variants. These variants can be visualized with an onboard instance of the integrated genome viewer within EPI2ME. The beauty of EPI2ME's human variation workflow is its flexibility combined with bioinformatic power. Investigators can select to examine only the variant modules that are relevant to them. There are only two required inputs, base called reads and the reference genome. However, you can further tailor this analysis by examining only targeted regions by providing a target bed file. This can reduce processing times, which can be especially important for time-sensitive analyses. All of our pipelines are open source and all defaults are built in for established best practices for our technology, but they are configurable if you need to adjust based on your own application. If we dive deeper into the bioinformatic analysis, two data files are required for the input into the human variation workflow. These include your sequence data, which can be in FASTQ or BAM format, and a reference genome file. For human samples, these must have the format of either UCSC or ensemble chromosome names. There are options to customize your genome, but for standard human samples, the workflow will check for the expected chromosome names. For this discussion, we're going to assume that you are inputting unaligned data. Following the initial input checks, the workflow performs alignment to the reference genome. Quality control and alignment statistics are output into an easy-to-read report, along with the expected aligned BAM files that are tagged with important information for each read, including mapping location and quality, modifications, and haplotypes. Based on the modules you select during setup, the workflow will proceed with variant calling using state-of-the-art variant callers. The workflow uses Claire3 to call single nucleotide variants. These variants are used to phase reads with WhatsApp and call haplotypes. The phase blocks are used to establish haplotyping of all other variants, including modifications. Following phasing, structural variants are called by Sniffles 2. Further, short tandem repeats are called with Straggler, and copy number variants are called by Spectre. These variants are then annotated according to the known genomic features for the standard human genomes. This is a key step as it streamlines the interpretation of nanopore sequencing data by linking variants directly to human-readable genome features. This is reflected in the variant reports. One report is output per workflow module, along with the functional bioinformatic outputs, including bed files, VCF files, and alignment files. As a bioinformatician, I appreciate that EPI2ME's human variation workflow outputs analysis results that are both high-level and very granular. These granular outputs include alignment files in BAM format, variant calls in VCF format, with and without clinically relevant annotations. This is important because these files can be directly ported into downstream analyses in your research setting to select relevant genomic regions to visualize and communicate results. Summary files for variants, alignment, and modifications are also output. These files are tab delineated and can be visualized as a table in your favorite table-based software, or, more efficiently, further processed and parsed downstream. Finally, a modification summary can be used to examine differentially methylated regions, methylation motifs, and segmentation analysis. Conveniently, these summaries are all reflected in the module reports. The tables and plots are interactive and allow you to directly examine your data. Now that we understand the approach of the human variation analysis, it is important to explore the expected performance of the Nanopore platform. From a single PromethION flow cell, it is possible to investigate the human genome at around 30x and get high-quality genomics and epigenetic variant from a single run using high-accuracy base calling. These include extremely precise and accurate single nucleotide variant calling with an F1 score of 0.997, which translates to a variant calling accuracy of 99.7% when compared to the ground truth. Nanopore methylation detection at a single base resolution is over 99.5% accurate. This is more accurate and sensitive than bisulfite sequencing. Structural variant calling in nanopore datasets reaches the ceiling of over 96% accurate. This is the gold standard in the field, as the quantification of structural variant precision and recall is limited by the available truth set established by other, less sensitive orthogonal technologies. And finally, we can see that the human variation analysis will output megabase scale phasing. When single nucleotide variant calling is considered, these include substitution variants, and indels. We also need to understand the computational limits of variant calling. On nanopore platforms, SNV calling is comparable to short-read sequencing in the number of true positives detected. Further, SNVs can be identified de novo without prior knowledge of location. In the genome viewer example, a G to A missense mutation was identified and is linked to Leber's optic atrophy. SNVs can be phased to provide maternal and paternal alleles and haplotypes, independent of trio sequencing. Processing these samples with nanopore sequencing improves the resolution of haplotypes in a way that is not possible on short-read platforms, with special emphasis on very similar genomic regions. In the example, reads mapping to the CFTR gene have been visualized. Mutations in the CFTR gene cause cystic fibrosis. Here, exons 4 and 5 of the CFTR gene have been phased to demonstrate that the donor sample is a compound heterozygote without trio sequencing. Haplogroups are directly tagged in the epitome human variation outputs. Structural variation has been historically incredibly difficult to resolve on traditional short-read platforms. Indeed, the measured accuracy of structural variant calling is limited by the lack of comprehensive truth sets established by orthogonal technologies. Structural variants include deletions, insertions, duplications, inversions, and translocations that range in size from 50 base pairs up to about 100 kb. On the Nanopore platform, structural variants are detected at 96% accuracy and, importantly, can be phased based on the surrounding single nucleotide variants. Duplications, deletions, and breakpoints can be easily identified with the resolution from long reads. Copy number variants include deletions and duplications that range from about 100 kilobases up to the megabase scale. It is possible to detect copy number variants at low depths, while phasing and breakpoint identification occurs at higher depth, for example, 30x. Low coverage copy number variant analysis can be useful for pre-screening a sample to examine any chromosomal level variations to select samples for downstream whole genome sequencing to enable more in-depth analysis. One of the most exciting applications of nanopore sequencing in the human genome is the characterization of variation in challenging genomic regions with complex structural variants and repeats. Nanopore data can resolve these particularly challenging regions, with one example being SMN1 and SMN2, which is linked to spinal muscular atrophy. The only difference between these two regions is a single SNP zoomed in on the right. Nanopore phase blocks enable the examination of this defining SNP to characterize SMN1 versus SMN2 haplotypes, as well as the identification of a non-functional pseudogene. In reference samples, typing was consistently possible on 10 out of 10 samples tested, sequenced at 30x with 15 kilobase read length. The detection and quantification of modifications in the human genome is straightforward in nanopore data. No additional library preparation is needed for detecting modified bases on nanopore data, nor proxies such as bisulfite conversion or deep sequencing kinetics. Nanopore methylation data spans the whole genome. Nanopore detection of modifications is accurate with low coverage as it relies on the direct detection of signal rather than a pileup, and at higher coverage can be phased into haplotypes with high resolution due to the long read lengths. Modification detection is controlled in Minnow during sequencing, and investigators can simultaneously detect multiple compatible modifications. Modification data can then be quantified by EPI2ME's workflows. Indeed, EPI2ME's human variation workflow outputs summarized modifications even across long repeat expansions, such as in the FMR1 gene. In the example, a disease phenotype was identified despite established heterozygosity of the repeat expansion length. Modification quantification of the locus revealed that high levels of methylation at both alleles repressed expression of the FMR1 gene, which is associated with a fragile X phenotype. Another example is Prader-Willi syndrome and Angelman syndrome, both caused by aberrant genomic imprinting in a known CPG island of the SNRPN gene promoter. A defect in methylation at this site causes a syndrome, but the methylation status determines which syndrome. Only the methylated allele is observed in patients with Prader-Willi syndrome, whereas only the unmethylated allele is observed in those with Angelman syndrome. With Oxford Nanopore, we can assess which is the cause of the appearance of the syndrome, as well as detect methylation and differentiate between the genomic abnormalities in a sample. For the final section today of my talk, we are going to do a demonstration of the human variation workflow at EPI2ME. Once you have the EPI2ME application installed on your computer, you are greeted with a home page. The application will ask you to log in with your Nanopore account to access previously run analyses as well as the cloud-based functions. The top section gives you an overview of the health of your system. The middle pane shows past analyses, and on the bottom, you can navigate to the EPI2ME blog posts, which might be helpful in your analysis planning. You next navigate to the launch pane on the left and scroll down to the human variation workflow. There are icons for the local installation or cloud availability. Today, we will be running a local analysis. To begin, you click Launch and under the Run Locally tab on the right. This brings you to the configuration step. Here is where we select the main workflow options. I am selecting all the relevant variation modules to run today. I further navigate into the main options tab and input information such as the desired sample name. In this case, I'm going with February and demo. I select my input BAM file from a place on my local system. And I also select my desired reference genome file. In this case, I have lowered the input data minimum required coverage because I subset the input data to have about 3x coverage to reduce the analysis time for this demo. I also wanted to perform phasing, so I turned that option on. There are options to configure parameters for the specific variant calling modules, but in most cases, it is best to leave the defaults as they are configured for the best practice for our platform. Please remember that these workflows are all generally configured to conform to our best practices. The only thing that I'm going to do now is change the number of available CPUs and memory that is available to the workflow during operation. This is because my instance for my analysis machine is pretty beefy, and so in this case, I'll have the ability to increase it. This is under the Nextflow configuration, and I'm going to increase to 46 CPUs and 180 gigabytes of RAM. Now we are ready to launch the run. We can start the workflow with a click of a button. Immediately following the launch confirmation, which is a pop-up window, you will see the run page appear. On the top, you can see the run status. Here, it is showing blue for running. Other options are red for error and green for completed. Within a few seconds of starting the run, you will see the jobs from the workflow initialize. Their status is indicated by bars on the screen. To view the standard log of the workflow, you will need to click on the Logs tab, which is up on the top left of the run page. Again, you will see the submission of the jobs via their standard names that are defined in the workflow code. In the panel underneath the log, you will see the workflow parameters that were configured during setup and some that are set as default by the configuration file of the workflow. Output files are listed as they are generated on a different tab called Files on the top left. Once a run is complete, you will see the jobs all under the progress bar go from running to completed. Sometimes a job might fail and be automatically resubmitted, which is a key feature of configured EPI2ME workflows. You will be able to view reports under the Reports tab. In this case, we are looking at the overall alignment report. The top pane contains overall statistics, and below that we can see some interactive graphs that are library-wide. These include read quality and read length and are all zoomable, so you can look at different populations of your reads. In this case, I'm going to zoom in on the accuracy graph to show you how you can interact. This is useful as these graphs are generated directly from the output data. Genome-wide library mapping is also interactive. You can navigate to different chromosomes and zoom in to see their coverage across each chromosome, delineated by color. So in this case, I'm going to zoom in. So we can see the chromosome coverage is less than 10 as I intended for the subset data. You can navigate to different parts of the report at the top right panel on each individual module report. This will jump directly to the information that you need. You can also go to different reports and different sections depending on what your interest is. On the top left, you can navigate between different variant reports. We can see the copy number variant report, which is similarly contains statistics and annotated CNV tables. Next, the small variant report includes substitutions and indels, as well as relevant statistics that can be reported based on your sample. This also includes ClinVar annotations in each report for the small variant module. Short tandem repeat expansions in the STR report will be classified by pathogenicity and annotated in the genotyping results. And finally, the structural variant report will have statistics, again, and a genome-wide view of structural variant events, as well as a quantification of the distribution of structural variants and their size across the whole library. A very useful recent addition to the human variation workflow is the inclusion of the integrated genome viewer under the Viewer tab. This allows you to interact directly with the genome to view detected variants. You can zoom in and out and see different annotated variants across your whole genome. You can navigate between chromosomes to see the different types of variants that are called. At the top left, you can navigate to different chromosome location. You can also mouse over these variants and get specific important variant statistics. In this case, I'm going to look at a CNV that's called on chromosome 16. You can see the type and the quality filtering that are output directly by the variant caller. These pop-up windows are very important to look at variants of interest. Thank you so much for your time today. With that, I'm now going to hand over to our partners who will present the variant interpretation tools that have been optimized to seamlessly integrate with EPI2ME. First, we will hear from Suzanne Drury, Field Application Specialist at Geneyx, followed by Samuel Strom, VP of Clinical Operations at Fabric Genomics.
Hi, my name's Susie Drury and I'm a Field Application Specialist at Geneyx. And today I'll be speaking to you about making the most of your data and the analysis for the future of clinical genomics. Just a brief overview of the Geneyx analysis platform. We support everything from raw data processing, the integration of flexible workflows, including customizable protocols, filters, and gene panels. You can perform reporting within the system, either automated or customizable. And we also support a series of data integrations via APIs, both to ingest your data to begin an analytical workflow, and also as a means of integrating your reports into sort of your electronic health records. Now, the system is extremely flexible and allows everything from black box testing, where you want to do sort of very high throughput, very automated testing, all the way through to white box testing, where you really want to be able to do a deep dive and sort of thorough analysis of your data. Now, Geneyx have a relationship with Oxford Nanopore. We're very proud partners. And as part of this, we have an integration with the Oxford Nanopore Cloud, with the EPI2ME platform, enabling users to go straight from that secondary analysis directly into the Geneyx tertiary analysis platform. In addition, if customers are utilizing their own secondary analysis tools, we also support the upload of the multiple VCF types that are generated as part of those secondary analysis tools as well. Within the Geneyx analysis stack, the VCF files will then be annotated with a series of data annotations, and then within the Geneyx platform, users can perform the interpretation and report generation. Now, one of the things with long-read sequencing and whole genome sequencing in particular is obviously the huge amount of data that's generated, probably in excess of 5 million single nucleotide variants and 25,000 or more structural variants. And now, particularly when we are thinking about assisting patient diagnosis, it's very important to be able to analyze your data quickly. And we've implemented a smart filter, which enables an enhanced annotation time, removes background noise, and improves focus on relevant variants. And we're doing this essentially by using a bed file to define a region for analysis, and then allowing customizable thresholds for CAD, for population frequency, and various filtering fields to enable sort of a fine balancing of retaining relevant variants whilst also reducing the background noise. Now, there are a number of long-read features which we implement within the platform, and I'll give you some examples of these in a moment. But you can look at phasing information, methylation analysis, repeat expansion as well, and we are allowing the upload of multiple different types of VCF to represent those different variant types. If we think about phasing initially, then in this particular example, we have a family which has a family history of a cardiac defect. And here we have an individual who was sequenced along with their parents, initially with short-read whole exome sequencing, and this identified a GDF1 variant, is actually known pathogenic variant, and it was maternally inherited. Now, the GDF1 gene is associated with two different modes of inheritance, both dominant and recessive. And the team wanted to sort of investigate whether or not there was actually a second variant that had not been detected via short read. So the proband underwent whole genome sequencing via Oxford Nanopore, and this identified a second variant in the GDF1 gene, and because of that use of phasing information and the display of phasing blocks, along with which allele each of these variants are present on, it was determined that this particular case had an autosomal recessive condition, this right atrial isomerism. Now, short tandem repeats are another variant type that are detected via long-read sequencing. And in this example here, we've got 16 individuals who had a confirmed pathogenic expansion via other methods initially, but were ran through Oxford Nanopore whole genome sequencing. And here we can see the repeat genotyping by the long-read whole genome sequencing, where we've got the expansion displayed in both allele 1 and allele 2. Now, within Geneyx, we display the short tandem repeats information for both of those alleles. We also have introduced a color coding system that indicates the clinical implication of the short tandem repeat, where we have the sort of the pre-mutation expansion ranges are indicated in orange and the mutations in red. You can filter based on these annotations as well. And we also show an in-house distribution of repeats. So here you'll be able to see within your own data set the repeat allele size distribution across all your different cases. We also enable the visualization of methylation along with your BAM file, and you can also do comparative methylation. So here at the bottom we can actually see that we've got the methylation data from 2 different samples and can visualize that differential methylation across the samples. I also wanted to touch upon the Geneyx phenotyper, and this is where we're using a gene phenotype association using either HPO terms or free text assigned to the patient, and we're using this to rank variants. And importantly, this is evidence-based. So I just have a brief image to show you here. So here you can see on the right-hand side, we've got our phenotyper score. I've sorted now the variants based on the score, and here we have the 2 GDF1 variants that we initially saw earlier in the presentation. And I can click on that information, and here I get all of those pieces of evidence that are linking this gene with the phenotypes that are assigned to the patient, and I can also link out to those various sources of information as well. This is a recent publication from a team in Leuven who have conducted Oxford Nanopore whole genome sequencing, and then as part of the publication they've also looked at the Geneyx phenotyper. So here you can see 20 positive cases and the ranking of those variants using the Geneyx phenotyper, and you can see that in 15 out of the 20 cases the known diagnostic variant was actually ranked top of the list, and in all but one case it was in the top 4. And there was only one variant that wasn't ranked, and that was because it was a mosaic variant and actually had not been detected by the secondary analysis part of the process. Just to sort of summarize then some of the Geneyx key features, we can analyze both short read and long read data for the same case in one platform. So this is obviously sort of supporting those customers who are starting to transition from short read sequencing and maybe implementing long read sequencing as that kind of that second tier approach, perhaps for those sort of exome negative cases. As I mentioned before, we can support the multiple VCF types. You can analyze your phasing data, your short tandem repeats, and your methylation, and making use of all of that in combination as well with that variant-driven prioritization. We didn't touch very much upon this because there's not sufficient time, but just to show you, this is how we implement different protocols within the system. So here you can see you've got long read sequencing analysis protocol along with an exome sequencing protocol, just for example. And within the system, you can fully customize your filters and your gene panels. We have a sort of a case management workflow. We also integrate auto-ACMG for single nucleotide variants and for copy number variants. And we have extensive reporting capabilities in the system as well. We can also support multiple clinical scenarios, so sort of germline and somatic, and also sort of carrier screening and pharmacogenomics as well. So hopefully that's just given you a flavor of how Geneyx really helps to make the most of your data as an analysis tool for the future of clinical genomics. Thank you.
Thank you very much, Susie, for this very interesting and informative presentation. Now, I would like to welcome Dr. Samuel Strom, VP of Clinical Operation at Fabric Genomics. Samuel will present how the seamless connection between EPI2ME software and Fabric Genomics platform enables rapid, high-complexity genomic analysis. Over to you, Sam.
Thank you, Cora. Hi, my name is Sam Strom. I'm a molecular genetics lab director and VP of Clinical Operations at Fabric Genomics. I'm excited to be talking to you today about how we are partnering with Oxford Nanopore Technology to deliver AI-driven clinical genomic insights. Before I go further, I do not have any conflicts of interest to declare. Fabric Genomics is a company based in the Bay Area. I'm actually down in Los Angeles. Hopefully I got to see you in my hometown for ACMG last week. But this company is based on using artificial intelligence and machine learning for genomic interpretation. Our collaborators and co-founder Mark Yandel and his laboratory at University of Utah have been using these approaches for over 10 years, and so we get to benefit from all that we've learned. We like to say that the AI era is finally catching up to where we've been over the last few years. So we work with hospital systems and commercial labs to deliver effective, high-quality testing in a rapid turnaround time. We do this with two different areas. So we have Fabric Enterprise, which is our software as a service. So this, you start with your genome data, and it processes it and delivers ready-to-interpret results. And then Fabric Clinical Services, which is my team, which is available to actually do the interpretation and reporting either in a collaborative fashion or as a standalone. So we have lab directors, genetic counselors, PhD scientists, folks who have a lot of experience in doing variant interpretation, curation, and sign-out. And we do this under the auspices of a CLIA and CAP accredited dry laboratory. So we do inspections, we have a quality manual, we have all the personnel documentation, we have everything that's needed to be able to do laboratory developed tests for sequencing for genomes, exomes, and panels. This is a great fit with Oxford Nanopore Technologies and what they're trying to do. So we've partnered over the last couple of years to be able to offer an end-to-end workflow for PromethION, EPI2ME into Fabric. It's a really smooth process that we've got down, working really well right now. The way we're able to really have this rapid turnaround time for the interpretation is leveraging two algorithms that we've developed, Fabric Gem and Fabric ACE. I'll go into more detail about Gem later, but to talk about ACE real quick, that's our automated classification engine. So this applies ACMG criteria to variants using gene-specific cutoffs and curations that we've done for over 2,000 medically relevant genes. Provides a lot of value there. So Oxford Nanopore technology can be done very quickly. I think all of you on this attending this webinar know this already, that one of the main benefits of this technology, in addition to long reads and other things, is that the library prep and sequencing itself is very fast. So on that top line, going from sample to variant calls and even through processing on Fabric takes about 1 day, maybe a little bit more, but in most cases it can be done in a day. And then that second day, my team has to wake up and have a cup of coffee and start interpreting results. Being glib a little bit there, we do actually have folks around the globe at different time zones so we can cover each other. But we do give ourselves 24 to 48 hours typically to do an interpretation and a final report so that we can actually do sample all the way to report within 48 hours. Which is pretty incredible as we kind of are. I'm reflecting about how we're about 25 years since the publishing of the human genome sequence. That took a very long time, many years to generate that first sequence. We can now do this in just 2 days, interpreted for that patient. I mentioned Fabric Gem earlier. This is the bedrock of our rare disease phenotype-based analysis. So this is an AI algorithm that identifies gene candidates. And the way that it does that, it's been trained on thousands of cases that have been reviewed by actual clinical teams for which variants are diagnostic for a given individual. It uses the input to train the model includes the HPO terms for the patients, so the phenotypes, as well as the disease gene relationships and modes of inheritance from various databases. It uses ClinVar and Nomad for ClinVar for pathogenicity for known variants, and Nomad for population frequency. And it also has a error model built in for the different platforms that we support. So for Oxford Nanopore Technologies, we were able to get a large data set of samples and train the model on the patterns of variants that occur in normal genomes and also the variants that are disease associated. So we're able to prioritize those genes that have genotypes, including recessive genes that have 2 variants in them. I'll show an example of that later. With phasing, we're really able to get down to a short list of candidates per genome that need to be reviewed to get high sensitivity. So this algorithm was published in 2021. This is now the old stuff, but even then, it was incredibly powerful and transportable. So in our original cohort of cases, we found that if we looked at the top 10 genes, we're able to identify the causal variants and genes in over 98% of those cases. In our validation cohort, we actually expanded it to a larger data set, and it performed slightly better, actually, than our original benchmark. So we were ticking up closer to the full diagnostic sensitivity. If you were able to see our talk at ACMG last week, we have some updates to this algorithm that are improving our sensitivity even further. We're now over 99% sensitive for the variants, it's with looking at an even shorter list of gene candidates. Another very exciting publication, this one came out more recently. This is just from the last couple of months. This is out of our collaboration with Justin O'Sullivan and his team in Auckland, New Zealand. So Auckland working with Oakland, say that 10 times fast. They benchmark nanopore sequencing on both cell lines and patient samples. This is a great read. I'm not going to be able to go through the whole paper in this span of time, so I'm just plucking out the part that I was most excited about, which is a real-world scenario where they used our pipeline from Oxford Nanopore sequencing into Fabric using the epitome caller for secondary analysis on 10 NICU patients. So these are babies that are born critically ill, immediately hospitalized, and are candidates for genome sequencing. Their team was able to deliver results in under a week for all patients, and most of them were done actually in 4 or 5 days, as you can see in that bar graph. And the diagnostic yield was 60%. You can probably do the math. They had 6 positive cases. And then retrospective analysis using more time and basically giving the researchers more time to see if they had missed anything combing through the data. There were no false negatives from the GEM reports. So this was a really effective, obviously it's a small sample size, but for these kiddos, they're getting a result really fast, and it's the right result. One of them is near and dear to my heart. So this is they was in the GBA1 gene. So I'm the chair of the variant curation expert panel for ClinGen for GBA1 for Parkinson's disease, which is dominant. This gene is also associated with autosomal recessive Gaucher disease. And indeed, what we saw in this patient was 2 variants, a pair of variants, a single nucleotide variant on one allele and a deletion on the other allele of 55 nucleotides. And the reason this is particularly exciting to me is the reason I got involved with GBA in the first place was that it's a very difficult gene to sequence using short read sequencing. There's a pseudogene that interferes and can cause both false negatives and false positives. And actually this single nucleotide variant that's detected is one of the more difficult variants to get right. And the 55 base pair deletion obviously is also difficult with short reads. And so it was amazing to see this come up through the Oxford Nanopore data, through Epitome, through Fabric, as just, yep, here it is. You got 2 variants, they're phased. We have one on allele 1 from dad, the other one's on allele 2 from mom. We didn't need the parents to know that these are 2 variants in trans that are causing Gaucher disease in this patient. So my team, the Fabric clinical team, is available to support your lab or hospital. We know not every group has enough capacity to do the interpretation and reporting, especially in such a time-critical fashion. So we're very excited to partner with Oxford Nanopore here because we feel that the democratized availability of the instrumentation, along with the high quality of the data that comes out with the long reads and just getting through pseudogenes and all that great stuff. But someone at the end of the day needs to interpret those results and deliver a report in a timely fashion. And so if that sounds like a good fit, we'd love to hear from you. So reach out to Oxford Nanopore Technologies or to Fabric directly if you're interested. We're starting to launch this rapid NICU workflow, this ultra rapid NICU workflow this year. So to summarize, GEM is our algorithm for AI gene and variant prioritization. It's peer-reviewed. It's published in Genetics and Medicine. We think it's an extremely high-performant tool with both high sensitivity and specificity. And we have updated it. We've updated the model to be trained on Oxford Nanopore data specifically so that we are robust to the different types of variants and patterns that we see in that data. Our clinical services are available. We would love to help you out to make sure that either to boost the capacity that you have or to give you capacity you don't currently have, if that's something you need to build out. So we are in a CLIA and CAP regulated environment for interpretation and reporting. And we have this NICU-ready, ultra-rapid workflow that can deliver results in just a couple days for these patients. So thank you so much for your attention today. And the last thing I just need to say for regulatory purposes that Oxford Nanopore Technology products are research use only. Any clinical or diagnostic testing that I'm referring to is all within the auspices of laboratory developed tests. Thank you very much.
Thank you once again, Jennifer, Suzanne, and Sam, and thank you everybody for joining us. You can find more information about today's broadcast using the QR code, which you can find on your screen. This knowledge exchange, along with all of the previous broadcasts, are available for you to watch on demand, so please visit the Oxford Nanopore website to check them out. You can also find more information about our upcoming webinar and knowledge exchange on the Oxford Nanopore website. If you have any further questions about your project or getting started with nanopore sequencing, please contact us on support@nanoporetech.com. Thank you all once again and very much enjoy the end of your day.